Where in the genome does DNA replication begin? Before a cell can divide, it must duplicate its entire genome so each daughter cell receives a complete copy. This makes genome replication one of the cell's most essential and tightly regulated processes. But replication doesn't begin just anywhere along the genome. Instead, it starts at specific genomic sites called replication origins, often denoted as ORI. From these origins, specialized molecular machines called DNA polymerases initiate copying, and replication proceeds outward in both directions. Think of it like reading a book. You don't start randomly in the middle. There's a specific place where reading begins. In the genome, the replication origin is that starting point. Identifying where ORI is located in a genome is a fundamental question in molecular biology. It matters not only for understanding how cells reproduce, but also for practical applications in medicine and biotechnology. One of the most important applications of understanding replication origins is gene therapy. Gene therapy uses genetically engineered mini-genomes called viral vectors. These vectors are modified viruses that can penetrate cell walls and deliver artificial genes into cells. In agriculture, viral vectors have been used to engineer frost-resistant tomatoes and pesticide-resistant corn. But the most dramatic applications are in medicine. In 1990, gene therapy was first successfully performed on humans. It saved the life of a 4-year-old girl suffering from severe combined immunodeficiency disorder. This little girl had been so vulnerable to infections that she was forced to live in a completely sterile environment. The idea behind gene therapy is elegant. You intentionally infect a patient who lacks a crucial gene with a viral vector containing an artificial gene that encodes a therapeutic protein. Once inside the cell, the vector replicates and produces many copies of the therapeutic protein, which then treats the patient's disease. But here's the key point for bioinformatics. For this to work, the vector must be able to replicate inside the cell. That means biologists must know exactly where ORI is in the vector's genome and ensure that their genetic modifications don't accidentally disrupt it. To understand replication, we need to go back to 1953 and the discovery of DNA's structure by James Watson and Francis Crick. They famously wrote, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." Their key insight was beautifully simple. The two strands of the DNA double helix are complementary. Adenine always pairs with thymine. Cytosine always pairs with guanine. This means that if you separate the two strands, each strand already contains all the information needed to rebuild its partner. Each original strand serves as a template for building a new complementary strand. The result is two complete double-stranded DNA molecules, each containing one old strand and one newly synthesized strand. This process is called semi-conservative replication because each new molecule conserves half of the original. Let's look more closely at how replication actually works at the molecular level. The process begins when the two DNA strands unwind and separate at the replication origin. This creates a replication bubble that expands outward in both directions. DNA polymerases, the molecular copy machines, then move along each separated strand and synthesize new complementary strands. They do this by matching nucleotides according to the base pairing rules. Adenine pairs with thymine. Cytosine pairs with guanine. The polymerases read the template strand and add the correct complementary nucleotide one at a time. This process is remarkably fast and accurate. In bacteria like E. coli, the polymerase adds about 1,000 nucleotides per second and makes only about 1 error per billion nucleotides copied. At first glance, replication might seem computationally trivial. Just represent the genome as a string and output a duplicate. But the biological reality is far richer. Replication requires precise coordination among many molecular components to ensure the genome is copied quickly and accurately. So how do we actually find the replication origin in a genome? This is where bioinformatics comes in. It turns out that the replication process leaves detectable patterns in the DNA sequence itself. By analyzing these patterns computationally, we can predict where ORI is located without needing to do expensive laboratory experiments. Identifying replication origins and understanding the patterns they leave in genomic data has become a key scientific and computational problem. It connects fundamental biology with practical algorithm design. In the upcoming lessons, we'll explore the specific algorithms and computational techniques used to find ORI. We'll learn about frequency analysis, pattern counting, and how subtle statistical signals in DNA sequences can reveal the starting point of one of life's most fundamental processes. That's our introduction to DNA replication and the problem of finding where it begins. You now understand what replication origins are, why they matter for gene therapy, how Watson and Crick's discovery revealed the copying mechanism, and how the replication process works at the molecular level. In the next lesson, we'll start developing computational tools to actually find these origins in real genome data. If you found this helpful, please like and subscribe.